Where we are, home stretch. I'd just like to start out by thanking Andrew and the SiteAb team, and maybe a quick round of applause. I think you're going to hear a very common set of themes here. I'll just touch base really quickly on what we know as the challenge, and then how Thermo Fisher has answered that call to try to make sure that we give you the highest quality reagents we can. I'll show you some examples of that, and then I'll just kind of land on how we've collected that data on our webpage so that it's easily accessible to you. So this could, I guess the alternate title for this is How to Buy an Antibody. So, you know, as we've talked about for the last 3 days, we know that antibodies are very critical for studying proteins and cell function, and lack of specificity can lead to a great deal of waste and frustration. Specificity verification can really help with that, particularly in an application-specific way. We estimate there are over 2 million antibodies on the market available from over 300 suppliers. That drive around $2 billion of revenue annually. Many of the antibodies are against individual targets, and that selection can be challenging and stressful, as we saw from that photo of Craig LaFlame. Sometimes these antibodies are unreliable, and hopefully the efforts that many of the vendors have been taking on will make that better. So we've adopted 3 major approaches to improve experimental reproducibility based on the International Working Group for Antibody Validation. And that starts with enhancing our own efforts to validate antibodies through stringent specificity testing, then increasing the visibility to that validation data to help you better select antibodies, and improving the reporting that we have around specifics such as clone, isotype identity, functional characteristics that help facilitate the replication of published results. So again, we look at selecting the right antibody for the right research needs. So we test these in at least one application. We look at either flow cytometry, Western blotting, CHIP, IHC, or ICC. We also do some IP mass spec, as Matt mentioned, and some SNAP chip assays. And we use a variety of specificity verification methods for that, either knockout, knockdown, cell treatment, or relative expression. In some cases, we do have more than one clone for the target, so I suppose in that case we also use multiple antibodies. Again, what's really important, I think, is finding the right clone for the right application. So as we determine what specificity tests to take, we think about what is the biological function of the target, what is its subcellular localization, what are the post-translational modifications that we might be looking for, is the gene essential, can it be knocked out, what is its turnover rate if we're going to try to knock it down, and in terms of the sample type, how does that influence the epitope exposure and the reactivity of an antibody for that specific application. All right, so now I'm going to just run through a few examples. So here using CRISPR knockouts, and we have generated, these are all generated using our lentiarray library. So we have 4 guide RNAs that have been specifically designed for every protein coding region of the human genome. And we use those to develop our knockouts. On the bottom left, you can see an example of a Western blot where we've knocked out beta-actin in, I think this was a HEC293 cell. We've got a wild-type control, a Cas9 control, and then the knockout that includes the guide RNAs, and you can very clearly see the absence of the band there. In the middle, we have a neurofilament target, and here we're doing an imaging experiment showing, again, a wild-type control, a Cas9 control in the middle, and the neurofilament knockout on the top right, and composite images on the bottom. And again, very clearly can see the disruption of the neurofilament and the absence of the signal there. And then on the far right, we have a flow cytometry experiment for a CD19 knockout. Again, a Cas9 control, a CD19 knockout. You can see the signal in the pink, and on the left, the negative control as well as the knockout. So, you know, certainly for genes that you can knock out and in cell lines where you have good expression, it's a very strong opportunity and technique. The second approach is to use RNAi. Again, in some cases, genes may be essential and they can't be knocked out easily. We expect to see loss of signal, at least, if not complete knockdown. These are coming out of, again, our own collection of siRNAs as well. And here what you'll see on the Western blot is an siRNA knockdown of STAT1. We've got untransfected, scrambled SI, and STAT1 targeting siRNA. And again, very, very clear diminishment of the band there. On the right, an example of an imaging experiment where we're knocking down ZO1 following, again, siRNA-mediated knockdown. You see a ZO1 knockdown and F-actin as a control, or not control, but just another marker, and the nucleus. And again, very clear knockdown in panel I of the ZO1 signal in these images. Relative expression is another common way that we look at the targets. So we're looking at cells that we know express a target abundantly versus another cell type that does not. So here we have a Western blot looking at pancytokeratin and A431, MC57, CACO2, and Raji cells. You can see an evidence of the band in A431, MC57, and CACO2, but not Raji1. Similar in the middle panel, we've got imaging with the same cell types, or some of the same cell types. So you can see the pancytokeratin signal in the A431 in panel A. In panel E, you see a negative in the Raji cell, and you can see that compared to the no primary control in panel F. And we can also do that in tissue. On the right, there's immunofluorescence in some tissue of mouse skin and mouse brain, where we expect to see a positive and a negative signal respectively. And again, very clear from those images that the antibody is showing specificity for that target in that application. All right, and lastly, another common approach we might take is cell treatment, where we expect a compound to have some impact on either the expression of a protein target or its post-translational modification. And in this case, we're looking at phosphoserine 345 on the CHEK1 protein. And you can see that when we add etoposide or hydroxyurea, we can get an increase in phosphoserine 345 on that target, and that's clearly shown in the bands in the Western blot. And that also shows up in the imaging on the right, where you can see the phospho-STAT1 signal in panel A, whereas you can not see it in panel E, where the cells are not treated. So again, very clear target specificity using compound treatment in both a Western blot and immunocytochemistry in this case. So how have we been doing with these efforts overall? To date, we've got about 12,000 antibodies that have gone through this advanced verification using different methods, diverse treatments, and various cell lineages. The numbers of antibodies by testing method are shown there on the bottom left bar graph. So relative expression was something we really kind of started with, and the knockdown and knockouts we've been a little more aggressively pursuing over the past couple of years. Compound treatment, about 2,000, and that other incorporates the IP mass spec and some of the snap chip assays where we're looking at epigenetic targets. Again, about 120 different cell lines, and the compound treatment's about 200 of those. So, making some headway. Obviously a lot more to do, but we feel reasonably good about the area that the advancements we've made. And these cover targets in a large swath of biology, so we do try to look at some of the most used targets and areas of biology that we think are most represented. So we've got targets in cancer, neurodegeneration, metabolism, cell signaling, and immunity, and you can kind of see the Venn diagram where some of these targets overlap pretty consistently across different pathways underlying cellular biology. We also try, to the extent that we can, to validate entire pathways. So this is an example of the EGFR pathway, and all the boxes that have a thick blue border around them are targets that we've validated using these methodologies. So we've been able to hit most of the nodes in this pathway, and I'll just show you one example of the EGFR receptor, which is starred there. So here we're looking at BCL2 first in panel A, where we've got a knockout, which we verified by Western blot. So again, you can see two controls, the wild type and the Cas9, followed by the inclusion of the guide RNA, very clear knockout of BCL2. In panel B, you can see a phospho-EGFR that we're able to manipulate using cell treatment in both an A431 and an A549 background using both agonists and agonist plus inhibitors. In panel C, you can see a GSK3 alpha beta that we knocked down with siRNA. Again, controls untransfected and scrambled, and then targeted against GSK alpha beta or alpha beta alone. And then finally on the right side, an imaging experiment where we're using cell treatment to phosphorylate GSK beta. And again, you can see here with the addition of colliculin A, we have phosphorylation of serine 9, whereas where it's untreated in panel E, you cannot see that signal. So when we go through these efforts, we highlight these with this very identifiable circle with a check mark inside on our website. That's what we call our advanced verification badge. And you can find that in the search results. And when you go to our web page, I'll just point out a few things here. This is a, I think, pretty sophisticated search page that allows you to filter on a number of different features of the antibodies that are important to you, including target species, clonality, et cetera. Under advanced verification, you can actually look at the types of methods that we've used, so you can filter on those methods as well. Additionally, I think Matt had mentioned to you the other day that right here we've got a new feature that shows the epitope. So when we know the epitope that the antibody is recognizing, we're calling that out for you clearly right there. In addition to the advanced verification badge, we also do try to highlight the number of published figures and references so you can get an understanding of how many other people have used the antibody and tested it. And there, in many, many cases, the antibodies have been verified in multiple applications. So typically, when you see an image down here, there'll likely be a series of images that represent either multiple applications or multiple specificities that we've used to validate the antibody. I think with that, I'll just land on a thank you and reiterate what you have heard today, which is if you're testing something that comes from us and it's not meeting your standard, just like the other vendors have mentioned, please reach out to us. If there are targets that you see on our website that haven't been verified, feel free to reach out to us. I've got Rachel Giesler's email on here. She's our current product manager for our antibodies, and you can feel free to reach out to her directly. You can also reach out to me if you like. Same email, david.piper@thermofisher.com. And, we're very committed to making sure that the community has reagents they can trust, and we appreciate the partnership with the other vendors, with SiteAb, and with the research community. So thank you.